Imagine you've got two bacteria sitting right side by side under a microscope.
Okay, just hanging out.
Right, just hanging out. And technically speaking, they are the exact same species. Like, they share the exact same fundamental biology. One of them is this completely harmless little laboratory workhorse, a strain called E. coli K12.
Right, the one researchers use all the time.
Exactly. You could ingest it and literally nothing would happen to you. But then the other one is E. coli O157H7.
Oh, wow, yeah. That's a bad one.
Yeah, that's the terrifying strain behind, those deadly headline-making hamburger outbreaks, the ones that cause severe kidney failure. So the big question is, what actually makes one microbe perfectly safe and its genetic sibling essentially an absolute killer?
It's such a fascinating contrast because it forces us to ask what a disease actually is, down at a biological level.
Yeah.
For a long time, we've just fundamentally misunderstood the relationship between microbes and humans.
And that is exactly what we are tackling in today's deep dive. We're looking through this stack of microbiological research, some really wild historical case studies, and evolutionary theory to figure out what actually makes a pathogen dangerous.
It's a really necessary discussion, I think.
Yeah, because we're going to unpack the specific biological factors that let these microbes cause disease. We'll look at how they get their microscopic weapons and shatter some really deeply ingrained misconceptions about how viruses evolve over time.
Because a lot of the folk wisdom we rely on, like the idea that pathogens always naturally evolve to become milder over time, or that being a killer is just, a permanent trait etched into a microbe's DNA,
Right.
It just doesn't hold up to modern scientific scrutiny.
So to make sense of this, we first have to separate a microbe's basic ability to cause harm from the actual amount of harm it ends up causing. When I was looking through the sources, I found it super helpful to think about this using a martial arts framework.
Oh, I like this. Walk me through it.
Okay, so think of pathogenicity as asking a very simple binary question. Does this person know martial arts?
Right. It's a yes or no.
Exactly. They either have the capacity to fight or they don't. So a microbe is either pathogenic, meaning it has the biological tools to cause disease, or it's non-pathogenic. It's a qualitative threshold.
You cross it or you don't. But virulence, on the other hand, asks a much more specific question.
Right, so if we establish that this person knows martial arts, virulence is asking, well, are they a white belt or a black belt?
Yeah, it's a spectrum. It measures the actual intensity of the damage. And in a laboratory setting, scientists don't just guess at this spectrum.
Yeah.
They use concrete metrics. Two of the most common ones you'll see are LD50 and ID50.
LD50 and ID50. So what do those actually stand for?
Well, LD50 measures the lethal dose. That's the exact amount of a pathogen required to kill 50% of a test population. ID50 is similar, but it measures the infectious dose needed to simply infect 50% of that population.
So it puts a hard, quantifiable number on the severity.
Exactly. But what is truly remarkable is how much our framework for understanding those numbers has evolved. Like back in the 19th century, early germ theory pioneers like Louis Pasteur and Robert Koch, they viewed pathogenicity as a fixed property of a species.
Like it was just hard-coded into them forever.
Right. The prevailing belief was that a specific microbe always caused a specific disease in the exact same way every single time. It was treated like this unchangeable barcode stamped onto the organism.
But modern microbiology paints a much, much more complicated picture than that.
Oh, absolutely. Fast forward to 1999, and microbiologists Arturo Casadevall and Lisa Ann Porofsky proposed this thing called the damage response framework, and it completely rewired how we think about infection.
Oh, so?
They argued that a disease isn't just an attack carried out by a microbe. Disease is actually defined entirely by host damage, and that damage, it can come directly from the microbe's toxins, sure, but it can also come from the host's own immune system overreacting to the microbe and destroying its own tissue in the crossfire.
Wow. So virulence isn't this absolute number living inside the bacteria. It's a relational outcome, like it takes two to tango.
Exactly. A perfect illustration of this from the research is the fungus Cryptococcus neoformans.
Okay, that's a mouthful.
It is, yeah. But basically, if this fungus enters the lungs of an immunocompetent person, so someone with a standard healthy immune system is practically harmless, the immune system just contains it, and the person likely never even knows they were exposed.
So the virulence in that specific interaction is near zero.
Right. But if that exact same fungus enters a host with a severely compromised immune system, like someone with advanced AIDS, It is highly lethal.
And the microbe didn't change at all.
Not one bit. Its
Oh.
genetic makeup stayed exactly the same.
Yeah.
The host context changed, which completely altered the virulence.
Which brings up, honestly, a logistical nightmare for early researchers. I mean, if virulence is so heavily dependent on the specific host and their immune system, proving which bug caused which disease must have been incredibly difficult before modern genetic testing.
It was a wildly chaotic era for medical science. I mean, before the 1860s, the dominant medical paradigm was the miasma theory.
The bad air theory, right?
Yeah, the belief that diseases were caused by bad air emanating from decaying matter. Correlation was constantly being mistaken for causation. The scientific community desperately needed a rigorous methodology to cut through all the noise.
And that arrived in 1884, right, with Robert Koch.
Yep, the German physician Robert Koch formalized his famous postulates.
Right, Koch established these 4 strict criteria to definitively prove a microbe is guilty of causing a specific illness. I think it's worth laying these out for the listener.
Go for it.
So first, you have to find the specific microbe in every single organism suffering from the disease, but not in healthy ones. Second, you isolate that microbe and grow it by itself in a pure culture in the lab. Third, when you take that pure culture and introduce it to a healthy, susceptible host, it must cause the exact same disease.
And finally, the fourth rule, you have to be able to isolate that same microbe from the newly infected host, proving it was the agent of disease all along.
It's honestly a masterpiece of biological detective work.
It really is. And Koch's physical rules dominated medical science for a century. But eventually, researchers hit a wall. Certain pathogens simply refused to comply with these rigid physical rules.
Right, the source material brings up leprosy as a great example. The bacteria that causes it simply will not grow in a standard lab culture, which immediately breaks rule number 2.
Exactly. And then think about a virus like HIV, which only infects humans. You obviously can't ethically fulfill rule number 3 by deliberately infecting a healthy human subject just to see what happens.
Which is wild because when the traditional framework failed or when the medical establishment just refused to believe the results, researchers sometimes resorted to extreme measures to prove causation.
Yes. And the most legendary example of this is the 1984 self-experiment by Barry Marshall.
Oh my gosh, this story is incredible. So picture the medical landscape of the early 1980s. The absolute consensus among doctors is that peptic ulcers are caused by excess stomach acid, brought on by stress or spicy food.
Everybody believed that.
Right. But Marshall, who is this Australian physician, alongside his colleague Robin Warren, they hypothesized that a bacteria called Helicobacter pylori is actually causing the ulcers.
But the establishment completely rejected the idea outright.
Because they thought nothing could survive in stomach acid, right?
Exactly. The assumption was that no bacteria could possibly survive the highly acidic environment of the human stomach.
So Marshall couldn't get the standard animal models to fulfill Koch's postulates. The medical community is totally dismissing his research. So what does he do? He takes a Petri dish containing a cultured broth of H. pylori, and he just drinks it.
He deliberately infected himself.
Yeah, he literally drank a beaker of ulcer bacteria, and within days he developed severe gastritis, which is a precursor to ulcers. He had his colleagues confirm it through an endoscopy and a biopsy, proving that the bacteria was thriving in his stomach and causing the damage.
He essentially turned his own digestive tract into a laboratory, just to physically fulfill Koch's third and fourth postulates.
It is such a remarkable moment of scientific conviction, and it paid off. Marshall and Warren eventually won the 2005 Nobel Prize in Physiology or Medicine because their discovery proved that H. pylori causes most ulcers and is a massive risk factor for stomach cancer. They single-handedly overturned 20th century medical dogma.
They did, but obviously, relying on scientists to chug bacterial cultures is not exactly a sustainable methodology for the future of medicine.
No, probably HR wouldn't approve that today. We needed a safer way to prove causation. And the research highlights a major pivot in 1988 when microbiologist Stanley Falcow introduced the molecular Koch postulates.
Right. This adapted the traditional rules for the modern genetic era. Falco argued that instead of looking for the entire organism, we need to look for the specific genes responsible for the damage.
So shifting from which bug did it to which gene did it.
Exactly. Falco realized that we shouldn't just be asking which bug is responsible, but which specific genetic sequence gives this bug its weapon. His molecular rules stated that a suspected virulence gene should be present in the dangerous strains of a bacteria, but missing from the harmless ones.
Like going back to our E. coli example from the start.
Yes, precisely. And to prove it, scientists would go into the lab and surgically delete that specific gene from the microbe's DNA.
Right.
If the microbe lost its ability to cause disease and then magically regained that ability when the gene was put back in, you had definitive proof.
You found the weapon.
You found the weapon. That realization shifted the entire field of microbiology. It proved that virulence is often not an inherent immutable property of a species. In a lot of cases, it's carried by discrete, movable genetic elements.
So once scientists knew to look for these specific genes, they started identifying the physical molecular tool that those genes were actually building. Because to actually cause a disease, a pathogen has to successfully execute 5 distinct stages of pathogenesis, right?
Yeah, 5 stages. It has to enter the host, attach to the tissues, evade the immune system's defenses, multiply, and ultimately cause damage.
And pathogens achieve these steps using a variety of specialized tools called virulence factors.
Right, so for attachment, they use adhesins. And rather than thinking of these as like generic grappling hooks, it's actually more accurate to view them as highly specific biochemical keys. These surface proteins are perfectly shaped to bind with specific receptor molecules on human cells.
Wow. Okay.
And then for the evasion stage, a lot of bacteria deploy capsules. These are thick, sugary molecular layers that surround the bacteria, essentially preventing the host's white blood cells from recognizing and engulfing them.
And then you have the toxins.
Right.
These are the proteins and enzymes that directly destroy host tissue. But the most surprising insight from the source material for me was that microbes don't always naturally possess the genetic blueprints for these weapons.
Oh, this is the best part.
Yeah, they can essentially rent them from other microorganisms.
The mechanism behind this is just fascinating. We're talking about mobile genetic elements. The textbook example here is Corynebacterium diphtheria, which is the bacteria responsible for diphtheria.
Right, and on its own, this particular bacterium is a completely harmless resident of the human throat. It lacks the biological tools to cause disease.
But that changes entirely if the bacterium is infected by a specific virus known as the betaphage. Now, a bacteriophage is a type of virus that exclusively targets bacteria. When the betaphage infects Corynebacterium diphtheria, it injects its own genetic material into the bacteria's DNA.
Okay, so the virus infects the bacteria.
Exactly, and this viral genetic code happens to include the tox gene, which contains the exact biological instructions for manufacturing diphtheria toxin.
So the harmless throat bacteria's own DNA gets completely hijacked and rewritten by a virus, turning it into a microscopic factory that just pumps out a lethal toxin.
Yep. The bacteria didn't evolve this weapon over millions of years. It acquired it instantaneously through an infection of its own. Pathogenicity was rented.
Rented pathogenicity. It fundamentally changes how you view bacterial threats. The capability to cause harm can just jump between organisms.
It really can.
But this molecular tool set also raises an interesting dilemma about the nature of the weapons themselves. If a toxin is just a protein engineered by a specific genetic sequence, context totally dictates whether it functions as a weapon of mass destruction or like a highly specialized tool.
Oh yeah, the dose and the delivery mechanism completely dictate the outcome.
Botulinum toxin perfectly illustrates this. It's produced by the bacteria Clostridium botulinum, and it is widely considered the most lethal substance known to science by weight.
It's unbelievably potent.
The lethal dose for a human by injection is incomprehensibly small. We're talking roughly 1 to 2 nanograms per kilogram of body weight.
Just to put that in perspective for you, a few hundred grams of perfectly dispersed botulinum toxin could theoretically wipe out the entire human population.
Yet medical professionals deliberately inject that exact same toxin into human tissue every single day.
Right, Botox.
Yes. The FDA approves the use of Botox, which is just a brand name for botulinum toxin for treating chronic migraines, severe muscle spasms, and, smoothing out cosmetic wrinkles.
It's wild. By utilizing microgram-scale doses and localizing the injection to specific muscle groups, the deadliest poison on Earth becomes a targeted therapeutic. The virulence factor has no intrinsic intent. It is simply a biological mechanism. The parameters of the encounter define its role.
So if these microbes can acquire such devastating tools so rapidly, and they can be so lethal, it naturally leads to a question about their evolutionary strategy. Why don't they just wipe us all out?
Right.
Or, conversely, there's this very common narrative that successful pathogens eventually evolve to become harmless to their hosts, because killing the host destroys their own habitat.
Okay, I am so glad you brought this up, because that is one of the most pervasive biological misconceptions of our time. Evolution does not prioritize peaceful coexistence. Evolution optimizes for a single outcome, and that is maximizing transmission to the next host.
It is entirely a numbers game.
Yes, completely. Evolutionary biologist Paul Ewald formalized this with his virulence transmission trade-off hypothesis. The most vivid demonstration of this actually took place in Australia in 1950.
Oh, with the rabbits, right?
Yeah, the Australian government intentionally released the myxoma virus into the wild rabbit population. Rabbits were this invasive species just devastating the agricultural ecosystem there. And the initial strain of the myxoma virus was incredibly virulent, killing over 99% of the rabbits it infected.
But researchers tracking the virus noticed a significant shift within just a few breeding seasons, right? The lethality of the virus dropped.
It did, but it didn't drop because the virus possessed some biological drive toward mildness. The original strain was failing evolutionarily because it was killing the rabbits way too quickly. The primary transmission vector for the myxoma virus was the mosquito.
Okay, so the mosquitoes had to bite them.
Right, and if a rabbit died in a matter of days, the window of time for a mosquito to bite that rabbit and carry the virus to a new host was incredibly narrow.
Oh, I see. So the strains of the virus that mutated to kill slightly slower kept the rabbits alive just long enough to ensure more mosquito bites. The virus essentially dialed back its lethality strictly because extreme virulence was impeding its ability to spread.
Exactly. In that specific context, lowering virulence aided transmission.
Yeah.
But if we look at a pathogen like cholera, the evolutionary math flips entirely.
Because of how it spreads.
Yes, the cholera bacteria induces severe life-threatening diarrhea in its host. That extreme virulence is not a biological accident, it is the core transmission mechanism. Cholera is a waterborne pathogen. The massive purging of fluids is precisely how the bacteria exits the current host and flushes into the local water supply, where it can then be ingested by hundreds of new hosts.
Wow. So for cholera, high virulence is evolutionarily favored.
It's required, honestly. And then on the furthest end of that spectrum, you have a pathogen like Ebola. When untreated, Ebola is horrifyingly lethal, but those outbreaks often burn themselves out.
Because the virus kills its host so rapidly that the infected individual becomes incapacitated before they can travel widely and spread the disease to a large population.
Exactly. From a purely evolutionary standpoint, killing the host faster than you can transmit to a new one is just a dead end. The mechanism of transmission is what dictates the optimal level of virulence for any given pathogen.
Which gives us a very, very different lens to view a recent global debate. During the SARS-CoV-2 pandemic, when the Omicron variant emerged, a massive media narrative claimed this was proof that viruses naturally evolved to become milder.
Oh man, evolutionary biologists were pulling their hair out over that one. They pushed back against that narrative hard.
Why? I mean, it did seem milder.
They pushed back because it represented a really dangerous misinterpretation of the data. The data did show that Omicron resulted in less severe clinical disease on average compared to the Delta variant, sure. However, that reduced severity was primarily driven by the fact that the population had acquired widespread prior immunity through a combination of vaccines and previous infections.
So it goes right back to the case of Duvall and Perevsky damage response framework we talked about. The microbe didn't intrinsically decide to become a white belt instead of a black belt. The host context changed.
Exactly. The evolutionary pressures selecting for the Omicron mutations were driven by immune escape.
Yeah.
The ability to bypass the antibodies the population had already built up and heighten transmissibility.
Yeah.
They were not driven by some biological mandate for mildness. There is literally no underlying biological law that prevents a future viral variant from achieving both high transmissibility and high intrinsic virulence simultaneously.
And understanding that evolution offers no guarantees regarding severity is exactly why anticipating these pathogenic shifts is a critical matter of global biosecurity.
Absolutely. When public health agencies conduct risk assessments for emerging threats, whether they're evaluating an outbreak of Mpox or, say, tracking H5N1 avian influenza spillovers in dairy cattle, they rely on this exact matrix. They evaluate infectivity, which is the capacity to spread, alongside virulence, which is the severity of the resulting disease.
So a pathogen exhibiting high levels in both categories represents the ultimate pandemic potential.
Yes. And this distinction between presence and disease also dictates how medicine is practiced on an individual level. A clinician has to determine if a patient simply has a microbe on them, or if an active damaging infection is actually occurring.
Like with staph, right? The research points out that roughly 1/3 of the human population carries Staphylococcus aureus in their nasal passages at any given time.
Right, it just acts as a harmless colonizer. Testing positive for the presence of the bacteria doesn't necessitate aggressive medical intervention unless the microbe-host interaction shifts and it begins causing actual tissue damage.
But the necessity of anticipating how these microbes might mutate brings us to a highly complex debate within the scientific community. If we need to know what a pathogen might do next, how do we study those future mutations before they actually occur in nature?
Right, which brings us to the core of the controversy surrounding gain-of-function research.
So, what exactly is gain-of-function research for someone who hasn't heard the term?
So gain-of-function research involves laboratory experiments where scientists deliberately alter a pathogen to enhance its capabilities, like increasing its transmissibility or its virulence. The objective is to proactively identify the specific genetic mutations that could give a wild virus pandemic potential, which theoretically allows us to develop targeted therapeutics and vaccines in advance.
And a major flashpoint for this debate occurred between 2011 and 2012, right?
Yes, a very famous case. Two separate research teams led by scientists Ron Fouchier and Yoshihiro Kawaka were studying the highly virulent H5N1 avian influenza. They modified the virus in a controlled laboratory setting to identify the exact genetic changes required for the virus to transmit through the air between ferrets.
And ferrets are used because their respiratory tracts are the standard biological model for approximating human flu transmission.
Exactly. And the publication of those studies triggered intense global scrutiny.
I can imagine. So what are the two sides of this debate?
Well, proponents of the research maintain that identifying these specific transmission mutations provides immense biosecurity value. They argue it allows global surveillance networks to monitor wild bird populations for those exact genetic markers, essentially serving as an early warning system.
But on the other hand, critics of the research argue that engineering a highly virulent virus to be easily transmissible between mammals creates an unacceptable risk. The argument is that an accidental release or some sort of laboratory breach could inadvertently spark the exact pandemic the researchers are working to prevent.
Right, and the debate over balancing the value of proactive knowledge against the risk of laboratory accidents reached such a critical threshold that the United States government actually instituted a formal funding pause on this specific type of gain-of-function research from 2014 to 2017.
Wow, just to step back and evaluate it.
Yeah, allowing time to rigorously evaluate safety protocols and oversight mechanisms.
It's a really stark reminder of the stakes involved when we begin manipulating the biological tools of virulence. Because, we have covered a massive amount of ground today. We established that pathogenicity is a binary threshold. A microbe can either cause disease or it can't, while virulence is the spectrum of how much damage it actually inflicts.
Right.
We explored how microbes don't always evolve their own weapons. They frequently rent genetic tools like toxins through viral infections. And most importantly, we clarified that microbial evolution is entirely indifferent to host survival. Its sole imperative is securing transmission to the next host.
Which is such an important takeaway. And honestly, shifting our perspective from eradicating the pathogen to managing the virulence opens up some truly fascinating avenues for the future of medicine. Traditionally, we've relied on antibiotics that are designed to kill bacteria outright. However, that intense evolutionary pressure rapidly selects for antibiotic-resistant strains.
Right, the superbugs. Which leads to the development of antivirulence drugs. So instead of trying to kill the bacteria, these new therapeutics are designed to simply disarm them. Like they biochemically block the adhesins from attaching to our cells, or they neutralize the specific toxins being produced.
Exactly. By targeting the rented weapons rather than the organism's core survival, we alter the evolutionary pressure. And the truly provocative question this leaves us with is, how will pathogens adapt to this new strategy?
Ooh, that's a good question.
Right. If we shift the biological mandate from survive this lethal antibiotic to find a way to infect a host without your primary molecular tools, what entirely new, unprecedented survival strategies will these microscopic organisms invent next?